Immunology live chat. My name is Colin O'Connor. I'm here at the AAAS headquarters in Washington, D.C. And today we're going to be talking about asthma. This is an issue that affects millions of Americans. It affects me. It might affect some of you in the audience. And to help us out, we're going to be speaking with Dr. Bruce Levy. He's the chief of the Division of Pulmonary and Critical Care Medicine at Brigham and Women's Hospital in Boston, Massachusetts. We'll also be speaking with Nandini Krishnamoorthy. She's an instructor of medicine at Brigham and Women's. Bruce and Nandini, hello. Thank you so much for joining us.
Hello.
Hello.
We're excited to be here.
I'm very excited to have you. So for those of you in the audience who are watching, if at any point you have any questions, feel free to leave them in the comments. We'll try to get to them later. So Bruce and Nandini, you have new research that came out on Friday, and we will get to that a little later. But first, Bruce, could you tell me in kind of general terms, kind of about who asthma affects, how many people does it affect worldwide or in the US, and what the symptoms are, what kind of issues it causes?
Thank you, Colin. And I just want to reiterate, I'm delighted to be here and excited to be with you and with our audience more generally on the Facebook Live event. So thank you to Science Immunology, AAAS, and to the Facebook Live folks for all their help with this event. So Colin, to directly address your question, it's thought that more than 24 million Americans have asthma, so you are not alone. This includes 7.4% or so of adults and 8.6% of children. It's the leading chronic disease in children, and it's the top reason why kids miss school. I prepared a few opening remarks to lay the groundwork here, and if it's okay with you, I'd like to just read through them for a couple minutes, and it will give the background that you asked for, as well as some information about how our study builds on that background. Great. So scientists still have an incomplete understanding of the origins of asthma, and there are no therapies currently available to cure it. There remains much work to be done towards understanding how this disease progresses in those who suffer from it. Adding to this challenge, asthmatic patients are not identical. Most commonly, asthma does start in childhood, and it can develop, however, at any age, and it affects all in terms of racial and ethnic backgrounds. Each patient has individual susceptibility to asthma triggers, and each patient has specific responses to treatment. Asthma is thus a syndrome, and it develops because of multiple factors, including both genetic susceptibility, it runs in families, as well as from environmental exposures. Because asthma prevalence over the last several decades is increasing, and our genetic code has not substantially changed during this time, external factors must be playing an important role in the observed increase in asthma prevalence. And so among these environmental factors are things like allergens, viral and bacterial respiratory infections, cigarette smoking, and occupational pollutants. So in recent years, asthma genetics and other approaches have really provided a lot of evidence regarding the background of this susceptibility, and it's profoundly affected our approach to asthma care. And advances in our characterization of airway inflammation, for example, have led to the recent FDA approval of new agents targeting specific inflammatory pathways that are important in promoting airway narrowing in patients with asthma. There's a particular form of asthma called severe asthma, and these are the most relevant patients for these new therapies, and severe asthma is debilitating. It's a syndrome that affects maybe 10% of asthmatics, and they don't respond to otherwise currently available therapies. So recently, to learn more about severe asthma, the National Heart, Lung, and Blood Institute sponsored a cohort study in which 111 children and 313 adults with severe asthma were followed carefully for 3 years. So relative to patients with non-severe asthma, those with severe asthma were more obese, had greater airflow limitation, as well as variable eosinophilic and neutrophilic inflammation. So for some among these patients, these new biological agents that inhibit eosinophilic type 2 inflammation hold great promise. But even so, new insights in severe asthma pathogenesis, particularly for patients with neutrophilic inflammation, so not only type 2, are needed. And this was the rationale for our study published last week in Science Immunology. So in our study, to model lung inflammation characteristic of uncontrolled asthma, we used a clinically common aeroallergen, house dust mite, in mice. And what we found is that when the mice were exposed to this dust mite, as well as to endotoxin, a combination not uncommon in dirty home environments or with an asthma exacerbation, this led to a phenomenon that was really exciting and interesting. It's called vital netosis. This was happening in the lung. So basically, netosis, N-E-T-osis, netosis is a dynamic cellular process of activation and release of neutrophil extracellular traps, also called nets. These are networks of extracellular fibers that are primarily composed of DNA from the neutrophils. This is coming from the nuclear component of the neutrophils that gets extruded as DNA and proteins. And this binds pathogens to kill them while minimizing damage to the host tissues. So there are two forms of netosis. One is called suicidal netosis, where the cell dies from this event. But there's this other process called vital netosis, and the cell can the cells do not die, but rather now these cells that are devoid of their DNA can retain many, although not all, of their cell functions. So these resulting cytoplasts are sometimes referred to as neutrophil ghosts. And these lung neutrophil ghosts were present in this model, and they contributed to the development and amplification of neutrophilic inflammation in response to house dust mite allergen. We also studied human patients with severe asthma, and interestingly, we saw the same features that were present in the house dust and endotoxin exposed mice: high neutrophil counts and evidence for vital netosis. So together, these new translational findings could be applied in a precision medicine approach for patients with severe asthma characterized by netosis. The mechanisms uncovered here are also likely to inform new therapeutic approaches to other forms of unrestrained neutrophilic tissue inflammation in the lung and elsewhere in the body. So Nandini Krishnamoorthy, who's here with me today, is the first author on this article. And together, on behalf of our co-authors, we're delighted to be here. We're excited to be here. Happy to take any questions that Colin or any of you in the audience have related to this or other areas of asthma research. And we thank everyone for tuning in.
Great. Thanks for that, Dr. Levy. And thanks for joining us, everybody who's joined since we started. We're talking about asthma today. If you have any questions, please leave them. If you have an experience with asthma or something that you'd maybe like to comment on, feel free to leave a comment about that. Let us know where you're watching from today. So one of the things you mentioned to me earlier, Dr. Levy, was that asthma's been around since ancient Egyptian times. We have documentation, I guess, from like 1500 BC about treatments for asthma. Why haven't we been able to kind of crack this nut yet? What is it about the disease that makes it so difficult to treat or cure?
Yes, thank you. It's a great question, and I think if we knew the answer, we would be on top of it. You know, it is a disease without a cure right now, and we see chronic inflammation, and it fails to resolve. So the normal restraints that you have on inflammation in the airway do not work well. And our current medications have some anti-inflammatory effects. So it can dampen some of the severity of asthma, and it can dampen some of the inflammation. And some people, in fact, most people find that this is quite helpful in their symptom control. But as I mentioned, folks with severe asthma, it's about 10% of the total. These medicines really don't quiet inflammation, and they end up having daily symptoms and a lot of disability, missed days at school, missed days at work. And so what we are focusing on in research moving forward is, in addition to anti-inflammatory agents, we're now looking towards natural processes that promote resolution. We're trying to look more closely and mechanistically at the resolution and repair processes, and to try to better understand why they're disordered and why they're not working well in asthma per se. But I wish we had the answer to your question, and we're working hard in the laboratory and in translational research to figure that out.
So when you talk about severe asthma, it sounds like not only is it more severe, but it's also kind of qualitatively different. There's something else going on at the cellular or immunological level. Is that right?
Yes, that is true, Colin. Many people with asthma will have an eosinophilic-rich inflammatory cell infiltrate, and it's characterized by what's called type 2 inflammation. And this is a very typical allergic type inflammatory response. In severe asthma, it's pretty clear that at least 50% of the people have either a mixed inflammatory response that's not purely type 2, or will have, frankly, a neutrophilic response, which is non-type 2 in nature. There are a very few percentage of people that will have a pauciimmune response as well, where it appears to be a disorder of the structural cells, like airway smooth muscle, directly, as opposed to the inflammatory response, which we see in most patients. So yes, severe asthma, even at the cellular level, and certainly in cluster analysis and expression analysis, we find very distinct inflammatory responses in your typical mild asthmatic patient.
Okay. Now, you've mentioned that there are a lot of different causes, and asthma plays out very differently for different people. Nandini, maybe you can speak to how you control for all those different effects in the laboratory, maybe what type of mouse models you work with. What does an experiment with asthma look like?
Sure. We have different mouse models, and as Bruce said, the idea of these murine mouse models is to recapitulate the human disease of asthma. And you have a range of severity that you see with patients. And what we are trying to do is mimic that scenario to the best possible ability with the murine model. And the advantage of using the murine model is you have extensive reagents. You can knock out a particular gene, which we have done in this particular paper. We have used the knockout mouse to confirm some of our results. And also you can overexpress a particular gene. So the murine model that we use for this paper involves methods to recapitulate the type 2, which is a very eosinophilic, or the most severe, which is the moderate asthma, or the most severe asthma, where we try to recapitulate high neutrophilia. So for the type 2, we use just house dust mite alone, which is a common allergen. It's in your mattress. It's just about everywhere in your house. And for the severe asthma model, or for the neutrophilic response, we use house dust mite, but also instill the endotoxin, or LPS, as is commonly known in the laboratory setting. So what happens is when you give LPS in the sensitization phase, it evokes a strong neutrophilic response. And in the absence of LPS, you get more of an eosinophilic response. So we were able to dissect that exposure to the endotoxin causes neutrophilia and the release of the cytoplasm. And it is indeed these cytoplasms that trigger high levels of IL-17, which is a cytokine or a pro-inflammatory molecule that can recruit neutrophils to the lung. So using these reagents, we were able to recapitulate the phenotype of moderate asthma and severe asthma.
So if I understand correctly, the endotoxin is something you would get from like a bacterial infection?
Absolutely. And there's also a lot of floating endotoxin. It's actually very difficult to get rid of endotoxin. It's in your house. Anything that's dirty probably has endotoxin.
Gotcha. So when you experience that in addition to an allergen, that's when you get this kind of severe, or at least in this case, this severe reaction.
Yes, and that is what we recapitulated and mimicked in our murine model. The murine models are not perfect of what we see in the human disease. Bruce, as a clinician, has already referred to the point, but it's a very nice model system to work with, and from where you can identify novel therapeutic targets, and that's what makes it an extremely valuable and critical tool.
Gotcha. So, Dr. Levy, I think you mentioned there's currently a higher rate of asthma in children than in adults. Is that right?
It's slightly higher on a percentage basis.
Okay.
Smaller lungs, smaller airways, a little bit more susceptible to airway narrowing.
Okay. It's not like it's becoming more prevalent. It's just shows up more in younger people.
It's increased in prevalence over the last several decades, but it's the direct answer to the question relates more to probably the size of the lungs and the airways than the prevalence percent. There's certainly more adults with asthma than children, but on a percentage basis, we see a slight increase.
I see. Okay. So how does this research relate to possible treatments for this kind of severe asthma?
So in a couple ways. One of them is that by identifying these cytoblasts, these neutrophil ghosts, and their relationship to interleukin-17A and neutrophilia, we have now biomarkers that we can take a large group of severe asthmatics and ask the question, which of these might have evidence for this process going on driving their uncontrolled asthma. So if we were to find evidence for these cytoblasts or high IL-17A levels, we might think about a treatment to either block the process of this vital netosis itself, or to just simply block interleukin-17A. Nandini referenced the fact that we could modulate this response in the mice by blocking IL-17. And so there's actually a clinical therapeutic that is already on the market and is being used in the treatment of psoriasis that blocks IL-17A. It's an anti-IL-17 antibody. And this is something that has been tried in asthma, but it was tried in all comers, in a broad population of asthmatics. There was no selection of patients for these specific mechanisms that are in this Science Immunology paper. And so what we could do now, though, is enrich our subjects to understand who has active vital netosis going on with high IL-17 levels, and now we could target that subgroup with this therapeutic that we know is safe and already available in the clinic. So this would help us do a very directed clinical trial and now potentially have a therapeutic for a group of patients that don't respond to other asthma therapies.
So there's a question in the comments which is related to something I wanted to get to, and this is, and apologize if I mispronounce your names as you write these questions. And Rajendra Shahi, she writes that inflammation is also associated with cancers. How often do you see people with asthma develop lung cancer? And that's something I wanted to talk a little bit about. Is asthma a risk factor for other diseases later in life, or are there comorbidities that you're also seeing that your research might be able to help with?
So, well, thank you for the question. We really appreciate it, and the engagement from everyone on the event. So, asthma is such a common problem that there are some reported associations out there with diseases, however, such as lung cancer, but there's no clear biological link there that's been robustly established, I would say. Certainly there are close associations with other forms of allergic disease. Eczema is one that comes to mind. And then there are some asthma triggers such as gastroesophageal reflux disease and sinusitis. And so there can be some associations there with those diseases as it relates to difficult to control asthma. So there are definitely associations with other diseases and other diseases of inflammation. However, when it comes to lung cancer, there's not a clear signal in the literature. That being said, asthma is so common that unless you look at very large populations, it can be a little misleading at times.
We have another question from Joseph Paris. Have you noticed any trends or changes in asthma incidence over the past few decades in the US or worldwide?
The main, thank you again for the question. The main trends are one of an increase over the last several decades. However, in this most recent decade, there seems to be some plateauing. This is a globally recognized disease. So this is not unique to either high resource settings or low resource settings, urban centers or rural communities. So this is a worldwide condition.
So what are the differences, and you mentioned this a little bit, in asthma prevalence and likelihood in the US versus other nations? I think you mentioned that the prevalence is about the same, but maybe the prognosis is different.
So the prevalence does differ in different regions of the world, with high regions in places like the US, the UK, Australia, and definitely some others, and lower, at least reported prevalence in some lower resource settings. But there's a belief that some of this may relate to underreporting, and not necessarily lower prevalence per se. So there have been a number of really well done epidemiologic surveys and related to the global burden of disease of asthma. And I would refer those tuning in to some of those studies. There's been a recent effort by folks here at the Channing Division of Network Medicine, led by Scott Weiss, to look at the relationship to vitamin D and the incidence of asthma. And there's been some very interesting findings by that group, as well as by Hans Bisgaard and the COPSAC group in Europe, linking vitamin D to incident asthma, and insufficient vitamin D being more associated with higher rates of asthma.
A question here from Abbas Youssef, and Nandini, maybe you can speak to this one. Can it be treated by suppressing specific cytokines involved in asthma through CRISPR-Cas9 technology? Is that something that asthma researchers are talking about these days?
I think when it comes to CRISPR-Cas technology, all researchers are talking about it. So definitely, but we have to be very aware of the fact that this is a multifactorial disease, number one, and just targeting a particular cytokine is not going to work. As Bruce mentioned in the previous question about treatments, we have to be aware of the severity of the disease, and we need to stratify the patients. And initially, it has to be tested in the murine model. There have been a lot of studies that are coming out where the CRISPR-Cas9 technology is working with human cells, but we have to proceed cautiously, and it cannot be just knocking down one cytokine, because, for example, interleukin-17 levels can regulate another cytokine like interferon gamma levels. So when it comes to knocking out in human cells, we have to proceed with caution. It cannot be something that we do with the mice, make a knockout and look at the effect, because some of these can have very dramatic effects and affect other organs.
We have another question here from Zara Mudaffar. What is the immediate therapy for this disease when the patient is suffering from difficulty to breathe? I imagine certain inhalers or that type of thing.
Yes, exactly. So thank you for the question. There are two types of asthma treatments. One of them is called symptom relievers, and the other one is called symptom controllers. And as the name suggests, the symptom relievers are what's used for immediate relief and bronchodilation to try to open the airways and make it easier for people to breathe with asthma. In the United States, the most common treatment for this is albuterol, which activates beta 2 receptors to lead to smooth muscle relaxation and a opening of the airway. Symptom controllers would be your anti-inflammatory, anti-leukotriene, biologic type therapies. Those often do not have an immediate effect within seconds to minutes, but with time, they will dampen the whole process. And so the symptoms become not only less severe, but less frequent as well.
I have a personal for me, not for you. Don't worry. But so since I was a kid, I've had a pet allergy. And I've always thought that asthma was kind of a different disease than the allergy. But is asthma kind of more accurately the symptoms of the constriction in the airways and the mucus and stuff?
So the pet allergy is a related process, I would say. Okay. So everybody who has environmental allergies do not automatically have asthma. And everybody with asthma does not automatically have environmental allergies such as to pets. However, there's a lot of overlap there, a lot of overlap. It's just not uniform, those statements. And in fact, the genetic susceptibility work that's been done has identified by GWAS quite distinct targets for allergy and for asthma, with the one overlap probably being the interleukin 13 locus. So the diseases are highly related, and a lot of the type 2 inflammation that is evoked by environmental allergies is similar to what many patients with asthma experience, type 2 allergic responses. But asthma is a broader syndrome than that, and so these are not entirely overlapping processes. So that all being said, if you are allergic to a cat and you get exposed to a cat and you get short of breath, you probably have not only a cat allergy or a pet allergy, but you probably have asthma as well. And we see this quite commonly. But it is also true that you might just get a rash and not necessarily asthma as your allergic response to the environmental allergen. So they're closely related conditions, but they are distinct.
Okay. So I kind of want
that. Yeah. Thank you for sharing your personal information.
Yeah. So this is a question from Amy Napoles, I believe is how you pronounce her last name. So what are your next steps for your research?
This is great. I'll start, and then I'd like Nandini to address it as well. It's pretty clear that this vital netosis process is a treasure trove of potential mechanisms that could be better understood by future research and also potentially leveraged by future diagnostics and therapeutics. And this, because it's a fundamental neutrophil activation syndrome, this may be quite relevant in the setting of many diseases, not just in the lung, but also elsewhere, because over exuberant neutrophil activation can lead to tissue damage. And one form of this is the netosis process. So we need to better understand the molecular drivers of that process and whether we could not only learn about pathophysiology and disease, but also about points of intersection that we might target with a new therapeutic approach. Nandini may want to also add to that.
Sure. One of the things that we discovered in our paper was that these cytoplasms or these neutrophil ghosts were able to evoke distinct CD4 T cell responses from the dendritic cells when compared to the neutrophils. So what are the mechanisms, molecular and biochemical, by which these neutrophil ghosts activate the dendritic cells, which are these professional antigen presenting cells that go and talk to the T cells? So this is one of the areas of research, and we couldn't answer it completely in this paper, but also it would give us novel therapeutic targets that we can identify on dendritic cells that recognize this class of cytoplasm or neutrophil ghosts, and help modulate the production of IL-17 or other cytokines. And as Bruce mentioned, the process of netosis is not restricted to asthma. In fact, it was discovered in so many other diseases as well, autoimmune diseases, infectious diseases. So modulating these responses would be very important.
Yeah, I'd like to also emphasize this point by Nandini because I think it's a really important one, in that a dendritic cell, when it sees a neutrophil, it didn't really respond differently than you might expect when a naive T cell got exposed to allergen. However, when that dendritic cell saw the cytoplast, the neutrophil ghost, it recognized this as a distinct process. You might think of it as almost an alarm signal, because by evoking a skewed immune response to generate IL-17, it recruits additional neutrophils to the space. So it's almost as if that cytoplast is carrying really important information that's recognized by the dendritic cell as, hey guys, we need some more help here, call in more neutrophils. And that Th17 skewing really helped to affect that.
I think it's like a vicious cycle, cytoplasm bringing the neutrophils, IL-17 productions.
Okay, there are a lot of great questions coming through, and I think we might need to start wrapping up. So if you still have questions, or if your question hasn't been answered yet, we can try to answer that with an extra comment a little later on. But for now, Bruce and Nandini, thank you so much for joining us. I learned a lot. I hope our audience learned a lot.
Thank you
all
very
for
much.
tuning in. Yes, thank you.
And thanks everyone.
Dr. Levy's and Dr. Krishnamoorthy's new paper, Neutrophil Cytoplasts Induce Th17 Differentiation and Skew Inflammation Toward Neutrophilia in Severe Asthma. That is in Science Immunology. It's live now. I think we're having some web page problems, so you'll have to access it later. But that is on immunology.sciencemag.org. Is there anything else you'd like to add?
No, just a big thanks, and we encourage people to read the paper, and if they have questions, to please shoot us a line.
Great. Thank you so much.
Thank you.
Bye-bye, everybody.